I'm going to do the bold thing and take out the microphone like a stand-up comedian or something. Hi, good morning. This is my first Monktoberfest, and it's been wonderful, so just thank you already in advance. So the title does give it away a little bit here, but I want to set the stage. So within your skull, you have an amazing bit of hardware. It weighs about 3 pounds. It operates at about 10 bits per second. And within that organ, you'll find the neurobullshit detector, an understudied and frequently underdeveloped piece of tissue. And my name is Ashley, and I'm a neuroscientist and educator. I'm a professor at UC San Diego, and I want to tell you a little bit about how to use this very important piece of equipment today. So, why am I here? A few things. You're going to find a lot of neuroscience information out in the world, and it's seductively creeping into everything. Everyone's slapping a brain on it. And I want you to have the tools to be able to suss that out. The second reason is that I've been teaching and writing about neuroscience for more than a decade, and somehow that hasn't solved everything. So I thought maybe I'd come to a tech conference and maybe you people with all of your tools could figure it out. And so I'm hoping that maybe I'll plant a little seed today that will go somewhere after this place. And the third thing is that, well, you have a brain and you probably want to know how to use it. There's an asterisk here though, because although you probably have a brain because you're here, you made it here, how much brain you have is actually unknown unless you've taken an MRI recently. So I want to give you something more to worry about today, which is that not that long ago, this is about 10 years ago, someone in China showed up to the hospital with like a little bit of slurred speech, not unlike the speech I heard last night a little bit, and showed up and had a huge chunk of brain missing. There's supposed to be stuff happening in the back here. So this person has what was called cerebellar agenesis. So they don't have a cerebellum, and they've never had a cerebellum, but it didn't show up until this woman was maybe in her 40s or 50s. It's not the only case of someone missing a significant chunk of brain tissue unbeknownst to them. And my favorite thing about this is the headline of this, which says 24-year-old pretty much okay without cerebellum. Like really excellent headline writing, right? So we're going to talk today about what I'm calling neurobullshit. And you've probably seen some of this out in the world already. There's, drinks that are meant to help your brain. You can buy a hair dryer that's called a neuro hair dryer. You could even get a med bed. I heard that that's a thing. So how do you know what is actually legit, right? So what we're going to do today is a little bit of what I call and what other educators call active learning. So I'm going to ask you for some input in a second. So I'm going to show you a headline, and I want you, on a scale of 1 to 5, or however many fingers you have, I want you to tell me what you think about these headlines. So we're going to do 3 of them. So the first one here says, brain implant translates thoughts to speech in an instant. Okay, so in a scale of 1 is bullshit, 5 is very accurate. What do we think? Oh, we got a wide range. We got a skeptical crowd here. Okay, I actually see a good range, but maybe a little bit more on the lower end of the spectrum. Okay, all right, we'll keep that in mind. Let's look at one more. So this one says brain cells that shape social choices identified. 1 through 5. Go ahead and give me a rating here. Another good mix. Wow, all over the place. Okay. All right, let's do a third. This one says, MIT proves ChatGPT makes you dumb. This is not the way I thought this was going to go. Okay, interesting, really interesting. Okay, great. This is why we do this, because now I know where you are. Okay, so what we're going to do here is we're going to work through each of these as examples. And, the whole scheme of this is to say, okay, in this landscape of headlines, how do you know what is actually real? So let's start with this first one. So brain implant translates thoughts to speech in an instant. Okay, so What we're working with here is a brain-computer interface. So this is a device that can take a brain signal, analyze it, translate into a message that can be carried out by some device external to the brain. This could be a prosthetic arm. We'll see it with speech in an instant. And sometimes these are called brain-machine interfaces or BMIs, right? There are several different kinds of these. So we can kind of put them in these three categories. So non-invasive, semi-invasive, and invasive. Coincidentally, the three different types of engineering management, right? And so on the non-invasive side, we have EEG and MEG. These are things that are not in your brain. They're listening to your brain. We'll talk about those in a little bit more detail in a moment. Then we can do semi-invasive. So I think it's funny that this is semi because they've taken this skull off. So it's pretty invasive. But in the grand scheme of things, it's in the middle. So this is where we've taken the skull off and you've put electrodes in, but you haven't actually like drilled into the brain. Or sorry, these are on the brain surface. And then invasive, where we've actually stuck electrodes into the brain. So brain-computer interfaces fall in these three different categories. But fundamentally, they're all doing the same process. So the first thing they do is they record brain or muscle signals. The second thing they do is that they process and decode, so they interpret those signals. And then the final thing is they output these to some sort of effector, like I said, an arm, a leg, a video game, whatever it might be. And in neuroscience, actually, this middle process has turned out to be one of the rare moments where this is easier than we thought it would be, right? This is not how it works in science. Things are always like, take so much longer than we think they're gonna take. And actually, as it turns out, this middle process of actually trying to decode what the brain is trying to say turns out to be a little easier than we thought. And I think that's amazing. So, this has been a long landscape of research. I'm going to tell you a little bit about it. But about 4 years ago, someone from mostly your world stepped into my world and made a big land claim here and said, oh, we're going to fix this whole thing. We're going to do it. You may know who this is, a guy by the name of Elon Musk, right? And he said the first Neuralink product is going to help someone control a smartphone without their thumbs, right? They're going to be able to think and do it. So they did some amazing things. So in 2021, they released one of their first videos, was this monkey playing mind pong, which is very cool. And the neuroscience community was immediately called to action, was like, wait a second. We have been doing this for a long time, and Andrew Schwartz said that kind of cortical control was demonstrated more than 20 years ago. And I don't know how many academics you know, but this is a shit post, right? Yeah, like we have done it. So shots fired, right? And so the whole neuroscience community was lit up, as was I. So I want to walk you through the history of brain-computer interfaces to make this point, right? Okay, so in 1965, Thelma Estrin, a woman with great arms, I will say, formulates requirements for a BCI, so makes this very formal. Here's what we need to happen in order for this technology to exist. A little bit later, UCLA begins work in monkeys, so as a first sort of non-human animal model to get this working. Then in the 1990s, we start doing work on humans, and you have this guy who's just having like the best day of his life, actually controlling a video game with his mind, right? 1990s. This is not 2021, 1990s, okay? 1998 is the first time we actually do this invasively in a human. Yeah, when Elon Musk was like a teenager. And then in 2000, we get the first formal definition of what a BCI is, and then finally we get the BrainGate clinical trial in the early 2000s. This is an amazing consortium. I'm going to show you one of their videos in a moment. And then in 2016, Neuralink is formed. Okay, so we've been here. We've been working on this shit for a long time, and a lot of this, almost all of this, is federally funded research that Elon Musk is capitalizing on. Yeah. So I want to show you a video because I I cry like every time I see this. It's amazing. This came out of UC Davis just earlier this year. This is a patient who has ALS, or Lou Gehrig syndrome. He can't speak. He's lost most of his motor functions. They've implanted a BCI in his brain, and you're going to actually see him speak for the very first time using this implant.
Demonstrations of BCI technology over the past few decades took weeks or months to get working. The first time he tried to use the system, it just worked.
Okay, ready?
That first time that he tried to speak and that word appeared on the screen, he started crying, I started crying, his family started crying. Basically everyone, we had to pause. We stopped the rest of that block of experiment trials. Everyone composed themselves. They were like, all right, let's do it again. And it kept working. So yeah, it was really special. It was a special moment.
At this point, we can decode what he's trying to say about 97% of the time, which is better than a lot of commercially available smartphone applications that try to interpret your voice, which hover around 95%. This is by far the most accurate decoding ever reported.
So it's not that we're reading people's minds. We're not detecting their inner thought or their inner monologue or even their their mind's voice. We're really detecting their attempt to move their muscles and to talk.
People who do not have the ability to communicate well or quickly are often isolated and lonely. Having something like this will give them hope and opportunity to be more of themselves.
So incredible stuff. And this is only getting better. The part of this that's an instant is actually more and more true. So this headline is about a different group doing similar work, and they really have shown that within like 20 milliseconds you can produce the word someone was trying to say. So it is almost instantaneous at this point. So believe it or not, this is very real and very, very cool. So how do we know it's real, right? You can see the outcomes for yourself. There's a video of someone doing this, right? It's right there for you. It was conducted by neuroscience researchers with experience. There's a visible trajectory for these ideas. You can trace this back to the 1960s. There's a history here. And finally, multiple groups have done this and are doing this, including in industry. And I don't want to like down talk the evolving nature of this in industry too, and that's amazing. I want both to exist, and I want credit to be given where credit is due. So very, very cool work here. So I'm glad to say that this does exist, and yeah, and we can stand behind it and be very excited about it. So let's go ahead and take the second one here then. So brain cells that shape social choices identified. Okay, so what is this about? So this is a little bit of a case of just some nice headline writing, right? So this is a case of something like this where, this is the paper headline. Oh, really fancy. It sounds really nice. I don't know if you can read the subtext, but it says, a neuron cluster in mice controls social choices and empathy, offering clues to autism and schizophrenia. So that all sound good. Okay, what is the actual paper behind this headline? Well, the paper is parvalbumin interneurons in the insular cortex control social familiarity and emotion recognition, right? A little less like spicy, okay? So in the middle here, this is like the job of a press release writer. They did a pretty good job here. Okay, but let's talk about what's actually in this paper then. So in order to do so, we gotta think about the different levels at which we can understand the brain, okay? So we're gonna start at the top here. We're thinking about behavior and cognition and networks. And when we think about this layer of understanding, we're thinking about what is it like to listen to the brain from outside, sort of like listening to a sports stadium, maybe the Red Sox and the Yankees, and you're hearing the crowd roar. And so you can get information globally about sort of, are there good things happening? Are there exciting things happening? Is everybody really disappointed, right? What is the tenor of the stadium? So that's our high-level understanding. Then we can go a level deeper. We can think about systems, circuits, and neurons. So that's kind of the equivalent of us going into one section. So this is my local women's soccer team, the San Diego Wave. They have a very gay supporter section. It's great. And you can go in and you can kind of hear what's going on in that specific section, right? So every time our keeper saves the goal, this section's roaring, and you can hear what's going on there. But then we can go a little bit deeper, get more specific information, right? We can listen to what single neurons are doing, maybe even what a synapse or a circuit or a channel is doing in this, right? And that's the equivalent of me seeing my friend on TV with his kid drinking a beer. And this is real. On national TV. So you get different information from the single person, then you do the supporter section, then you do the whole stadium, right? But all together, these are all each very important pieces of information in order for us to understand what's going on in the brain, right? But typically, neuroscientists are observing a particular window here. We're observing this within a particular context. And so, for me, like my PhD was in this level, sort of system, circuits, neurons. And usually that's because we don't have a choice, right? I have to kind of choose a level here. I can't measure all of these things simultaneously. So this is kind of where I would be. So this paper is very interesting, and it's an example of this. It's a specific context in which we're understanding something. So we need non-human animal models to get at this kind of level, and we need those models to get at mechanisms. And so when I say mechanisms, I mean things like how are different neurons in your brain connected? I mean, what are the roles of specific genes or molecules or proteins or even different circuits? And I mean things like, what's the causality in this system? Is there a necessary component? What components are sufficient, et cetera? So we, in almost every case, need a non-human animal model in order to answer these questions. And it doesn't even have to be an animal. It could be a batch of cells. It could be what's called an organoid, which is where you let cells just grow in a dish and they kind of turn into organs. It's very sci-fi, but very real. And this is a really helpful thing for us. So when we look at this paper, this is just that kind of specificity. It's a particular kind of neuron in one brain region, helping us understand a social behavior, but in this case in mice, right? And so we have to always ask, does the animal model tell us what we want to know in a human model? And that's always the question we need to ask ourselves. So we use these non-human animal models to create contexts, right? So those contexts are the genetic and molecular context, like for a particular disease, such as cancer or Parkinson's disease, that we want to study. We want to understand what happens when you tip this thing over, what else tips over in that system. And we especially need to do this, again, when we can't study those contexts in humans. Like I can't ethically give someone Parkinson's disease or give someone cancer, right? If I want to go into their brain, I need to have a context with which to study it. I wanted to give you a really specific example of sometimes where these contexts come from and how we discover them. And this one's just wild. So in the 1980s, this synthetic opioid was being used by some folks. It's called MPPP. And somebody messed up at some point, and instead of making MPPP, they made MPTP, which is a different compound. And a bunch of the folks who had used these drugs showed up at a hospital in Santa Clara with Parkinson-like symptoms, and they were diagnosed with Parkinson's disease. These are like young men mostly diagnosed with this, right? As it turns out, this drug, for reasons we don't fully understand, this drug actually creates a Parkinson's-like context in the brain. It kills the same exact cells that Parkinson's normally kills. So what we can now do, and what researchers do on a regular basis, is take this toxin, inject it into, say, a mouse or maybe a monkey, and then study what happens when you do, because that can tell us what happens with Parkinson's disease. So this is just one example of how we actually create a context. We use something sometimes completely sort of out in the world, which we didn't expect and we didn't plan for, and use that to understand something that we care about, right, which in this case would be Parkinson's. Someone wrote a whole book about this, in case you're curious, called The Case of the Frozen Addicts, referring to that group of people. So, all that said, obviously, non-human models are limited. There's only so much we can do with a non-human model, right? So once again, I'm going to ask you to kind of give me your opinion here. So in a moment, I'll have you vote. So just a classic thumbs up or thumbs down. So using only a mouse model, just mice, could we do the following? Okay, first one is determine the cause of schizophrenia. Thumbs up or thumbs down? All right, I see mostly thumbs down, and I would mostly agree. I think single-handedly, with mice alone, we're probably not gonna do this, right? We might get some clues, but we're probably not gonna single-handedly determine a cause, if there even is a single cause, which is maybe a bad premise in itself. Okay, second one, identify genes that contribute to schizophrenic phenotypes, or maybe the way that schizophrenia might show up. Thumbs up or thumbs down, how do we feel? Yeah, I agree. I think we've done this already. There's a lot of work on the genetics of schizophrenia, and we study those in mice, and they teach us things about what happens like in a neural circuit, for example, when schizophrenia happens. Okay, finally, describe cell types that encode perceptual differences. Thumbs up or thumbs down? Different cell types in the brain that could help us understand perception. A little more mixed on this one. This one I think is pretty good. I mean, mice have a different perception than us of the world. We have to take that into consideration. Like, it's hard to, for example, study hallucinations in mice. You can, there's ways to do this, but it gets a little tricky. But we could identify some cell types that maybe are involved in this process. So yeah, probably, we do this, right? So this paper is an example of maybe a mild hype headline, but pretty legit science, but limited in the scope that it can tell us about human behavior, but it gives us some insight. So once again, it was conducted by neuroscience research. There's a visible trajectory for these ideas, which I didn't talk about, but you can look at the paper and they'll show you that. And finally, there's some replicability here. So other groups have done this kind of research. Okay, with that, let's get into our third paper, the juicy one, right? So this is a paper I have been trying to avoid, in all honesty. However, it keeps showing up in my feed in like a number of different ways. I don't know if anybody else has seen this, right? So AI harms real people. MIT just completed the first brain study. This is shutting down your brain. This is like the stuff that's showing up on my, just typical Instagram feed. And this is the kind of information that's getting out in the world. I'm not going to go into a super deep dive here. I want to invite you, if you want to hear my wife and I talk about this for about an hour and talk about more than just the neuroscience, we did on our podcast called Change Technically. In this episode, You Deserve Better Brain Research, produced by Danilo, who's also here. So you can hear more if you want to listen to this, but let me sort of just give you a quick breakdown of what the neuroscience in this paper is doing. So first and foremost, this is a preprint, and preprints are great for science. We saw the importance of preprints during the pandemic, right? So bioRxiv has been around since 2013, but in 2020, we saw the most preprints on bioRxiv than we have ever seen because it was a rapid way to get information out about what was happening with SARS-CoV-2. There's other preprint servers. Archive, which I'm sure you're familiar with, has been around before that. That's where this paper came from. And medRxiv is for clinical information that came out a few years later. And preprints have really taken off. This data is only 2014 to about 2018. But if you were to look in 2018, you'd see like a crazy spike, like I said, going into 2020. So these are important, but there's a big, big but here, which is that they're not peer-reviewed. And the peer review process in science is not perfect, but it is the best thing we have at the moment for us to say whether or not science makes sense to other scientists. And so this is the major downside of preprints, and it's just the thing that we always have to think of with a grain of salt. So I'm here basically peer reviewing this paper here for you all, right? So what is the main neuroclaim in this paper? So the main neuroclaim, and there's lots of other claims, but we're gonna focus on the brain stuff. The claim is that participants showed weaker neural connectivity. And they say this in a number of different ways, but fundamentally, this is the thing they would like you to believe. Okay, so let's think about connectivity. And so to think about connectivity, we need to think about plasticity, because fundamentally, this is a claim about your brain changing when you use ChatGPT, right? So this is a question of plasticity. So how does plasticity happen? Well, the first thing I want you to know is that the structure of your brain doesn't change very much. Like the gross structure of it doesn't change very much. There are major exceptions to this, of course. If you have some very extreme form of dementia, for example, here you see advanced Alzheimer's, which we heard about yesterday. This causes dramatic changes in the structure of your brain, right? That's where you see big structure changes, not depending on whether you went for a walk or not today, right? That's not gonna do it. So most plasticity is not happening from big structural changes. It's happening because of the connections between your neurons, those synapses that are joining those neurons together. Or it can happen via the expression of your genes, right? So you have a genome, and the expression of that genome can change over the course of your life on some time scale. The thing is that we can't actually measure either of those things. We can't measure synapses in individual neurons, and we can't directly measure gene expression in living human beings. So we are fundamentally limited in our ability to measure plasticity of that kind. We can do things like you see on the Muse album cover here, which I love, it's like a neuroscience cover. We can do things like measure the tracks in your brain, the big tracks, but these are not single neurons. These are big bundles of neurons all tied together. So what can we do? Well, everything we do is not nature itself, right? So what we observe is not nature itself, but nature exposed to our method of questioning. I think this is a really fundamental idea in any biological study, right? So we're never looking at the thing directly, we're observing it through a window, right? So we're gonna observe the brain again through a window. And in this paper, they use this technique called EEG. And EEG, going back to our analogy of the stadium, is like standing outside of the stadium and listening to what's going on. It's a very coarse measure of brain activity. And EEG is a few centimeters away from where the stuff is happening. There's a couple things that make EEG kind of a difficult technique. And the first is just that you're not really up against the neurons themselves, the things producing the electrical current. You're like a centimeter or two away depending on the thickness of your skull. So you're already kind of far from the source. That's a problem. So we can't resolve single neurons in this technique at all. You're listening again to the stadium. So EEG activity looks something like this. You get these kind of squiggles. These are different channels, and stuff is going on over time. And we can do things like try to infer what's going on in between these channels. So we can infer if those channels are more correlated in time using some fun causality. We can ask, okay, these two channels are probably doing something together. Maybe they're connected. And we call that functional connectivity. So we can generate a connectivity matrix between our different channels, and then we can draw fun maps saying, okay, we think these nodes of the brain are connected based on the fact that their activity is correlated altogether. So we can do this in different conditions as well. Here's something that looks kind of similar to what I just showed you, a quote-unquote like normal neuroatypical brain. And here's what it looks like in epilepsy, for example. So in epilepsy, you see a ton of activity here, also a lot of synchrony, and so actually more functional connectivity in an epileptic brain. So the problem is with techniques like these is that it's not always clear whether more is better, right? So I just showed you that in an epileptic brain, there's more functional connectivity. If we think about something like size, there's some good evidence that like brain area increases in size when you like learn how to play the piano, when you get really good at basketball. There's this famous study with London cab drivers having like a big spatial memory part of their brain. So like there's some decent evidence that bigger is better in terms of the brain, but there's also some studies that show that if you overwork, you get increases in your gray matter. So what is that about, right? What does size actually do for us? Thinking about connectivity, I showed you that epilepsy causes increase in functional connectivity. But something also we can think about is that over the course of brain development, you actually lose connections. So you overproduce brain connections, then they get pruned back over the course of development. So in some ways, having fewer connections might actually be better, like more streamlined highway system, basically, in your brain. But then the claim of this paper is that using ChatGPT lowers your connectivity. So where do we place that, right? So at the end of the day, you need a theoretical framework. And just like we did with the BCI study, you need a development of ideas, a theory for why the brain is the way it is and what it means for it to be off its kilter, right? I just want to say, too, that as EEG headsets get cheaper, they get easier for people to just pick up and use and throw their data on their study and put developers under a study with EEG, right? And then show you the data, right? So this is just getting easier and easier. You can get an EEG headset for like $500 and do your own study and then claim that like, I don't know, productivity is linked to alpha or something, right? So my message to you is I want you to be really skeptical, because it's not always clear what like less means or more means or alpha means, etc. Okay, so the question shouldn't always be what's happening in the brain, right? I'm a neuroscientist, I love brains, but that's not always the question we care about, right? Sometimes the question actually needs to address directly what we care about. And if it's the case of ChatGPT use, well, then we might care about students cheating, or we might care about the ability to critical think, or whatever it is. That's the thing we need to study. And so, often that thing is human behavior. And so let's do the human behavior work instead of putting a brain on it, right? So this study didn't test ChatGPT leads to dumb. It didn't really demonstrate changes in neural connectivity. Once you get into the science of it, maybe, right? It's still hard to know what to make of these changes ultimately. So sometimes the best neuroscience research doesn't have brains slapped over it, happens in a non-human animal model, and isn't done at a prestigious institution, right? Most of the research I talked about in this talk was done in the UC system, which is near and dear to my heart. Okay, I'm hitting time, so I'm gonna skip past. If you wanna ask me about these things later, we can. Just other guidelines, don't trust fMRI research. Someone put a salmon into an fMRI scanner, we can talk about that. Brain regions are hardly ever responsible for a specific thing. And finally, very few things apply to all brains. So if you're a left-handed person, your brain is actually probably a little bit different than the right-handed people in the room in terms of where your language dominance is. So if you want to talk about these things later, come find me. I am online, and you can find me there. Thanks so much for your attention.